Hello, so this is my presentation on the cell cycle. The goal of this presentation is to learn more in depth about the role of various proteins in signaling pathways leading to cell proliferation and how mutations can ultimately lead to cancer. So the specific genes that I will be covering in this presentation is ATM and ATR, as they both work together hand in hand. So here to discuss the role of ATM and ATR. They're also better known as ataxia telangiectasia, and they are mutated by a member of the phosphatidylinositol 3 kinase family of PIKKAs. So what do ATM and ATR do in the cell? They basically maintain genomic stability in the face of constant exposure to exogenous and endogenous DNA damaging agents, and they both work as a signaling network. What exactly do they do? So ATR kinase activity is activated primarily in response to a different set of DNA lesions, ultimately leading to including those caused by UV light and in response to stalled replication forks. Once activated, ATM and ATR phosphorylate is an overlapping set of DNA repair and checkpoint targets. So they basically detect DNA damage, and they both work hand in hand. Now to discuss what's going on in this picture right here. So in step 1, ATM exists as dimers and multimers in which the active site is blocked. Double-strand DNA ultimately break. In step 2, this leads to an ATM intermolecular phosphorylation and the dissociation of the dimer and multimer and exposure of the kinase domain. In step 3, the ATM is then thought to be targeted to DNA lesions via adapters. And in step 4, the focus formation follows a phosphorylation of ATM targets, such as the PF3, as you can see right there, it is the red, and the cellular outcome will depend on whether successful DNA repair occurs. So now here to discuss about mutations in the ATM genes and to discuss its functions. So the main function of the ATM protein is that it coordinates DNA repair by activating enzymes that fix the broken strands, and efficient repair of damaged DNA strands will help maintain the stability of the cell's genetic information. So it plays an important role in the normal development and activity of several body systems, including the nervous system and, of course, the immune system. So the mutations here are germinal and somatic. The cellular phenotype of the AT will represent genome instability, deficient DNA damage, response DDR, and elevated oxidative stress. So there are various types of mutations that are going on. They have been dispersed throughout the gene, and therefore most patients are compound heterozygotes, which means that most mutations will appear to inactivate the ATM by a truncation or a large deletion. Patients with severe form of AT are homozygous or compound heterozygous for null ATM alleles. The corresponding mutations will ultimately lead to truncation of the ATM protein and subsequently to its loss due to instability of truncated derivatives. Overall, it causes this disease or disorder called ataxia telangiectasia, and it's also the cause of cancers such as bladder, breast, and melanoma. It's also important to know that people with this disorder will have mutations in both copies of the ATM in each cell. So now to discuss the ATR gene. Basically, the function of the ATR gene is that it maintains genome integrity by serving multiple roles in the cellular response to DNA damage and endogenous replication stress. It signals to regulate the firing of replication origins to the origins and the repair of damaged replication forks, and to prevent the premature mitotic entry. Mutations in the ATR gene are very, very rare and don't happen often. They are compatible with life only that is hypomorphic or heterozygous. There's a clear link that has been found between the disease and ATR gene mutation. It's called the Seckle syndrome. While ATR has been proposed to serve as a haploinsufficient tumor suppressor in some types of cell deficiencies, and its activation has been detected in most cancer chemotherapies. So basically, it has somatic and missense mutations, and this overall says that single nucleotide substitutions have been described in various types of carcinomas, and missense mutations have been found in the P13 and 4 kinase catalytic domain and in the fat domain of breast and lung cancers. There's also been some silent mutations, but not very popular. Some of the cancers that it causes are breast, skin, and overall affects your central nervous system. And there has been research that has found links to colorectal cancers. And it's important to know that ATR deficiency at the organism level will affect normal development, tissue homeostasis, and aging overall. So the question here is, can we live without ATM and ATR? The answer is half yes, half no. So ATM deficiency will overall lead to progressive neurodegeneration, and ATR deficiency will lead to autosomal recessive disorders. So you will have some brain damage, you will have some just overall nervous system disorders. And people who are heterozygous for ATM mutations have slightly increased incidence of cancers. So far, there has been no human diseases that have been associated with the total loss of ATR. However, it has been shown that partial loss of ATR activity has recently been associated with the human autosomal recessive disorder, also known as the Seckle syndrome. So by having deficiency of the ATM, you are more than likely going to have progressive neurodegeneration, immunodeficiency, predisposition to malignancy, and radiation sensitivity. So those are just some little clues to look for. And these are my sources. Thank you.